Okay, so we have the cranial fluid from the ventricles and the whole body of the central nervous system making its way back to the heart like the lymph does in the body, and comes out of the central nervous system and out of the ventricles into the subarachnoid space. And here we can see it going down the spine, around the spine. It also will be around the whole brain and the brain stem. But in this picture we've got the nerves coming out, and its way home is down through these nerves, make its way out of the nerves into the lymph and back to the heart. So it returns its journey. And we'll look at the cranial nerves later, because there's a slightly different thing involved with that. So the cranial fluid from the ventricles come out, and the cranial fluid from the body of the central nervous system. Now, there's a difference between the cranial fluid in the ventricles and the cranial fluid in the body of the central nervous system. The cranial fluid, as it makes its way out of the central nervous system, it's been used. The oxygen has been used up, the glucose has been used up, and carbon dioxide has been produced. The cranial fluid that is making its way out of the ventricles, it still has its oxygen, it still has glucose, it still has salt, so it's unused as yet. So what is the reason for this? Well, for me, the reason this hasn't been used is that it is for the nerves, the stretch of the nerve between where it leaves the central nervous system and where it goes out through the dural membrane. There is no blood supply to this, so it can't create the salt that it needs, it can't create the glucose that it needs, it can't create the oxygen that it needs, so it needs to get it from somewhere else. So the cranial fluid coming out from the ventricles, it comes out from a foramini in the base of the fourth ventricle, comes down in this subarachnoid space around the spine. This has this. It has the glucose, it has the oxygen, it has the salt. And this permeates through the whole of this subarachnoid space around the spine, and it's especially needed in the bottom of the spine where you've got this horse's tail, long lengths of nerves that don't have their own supply of oxygen, glucose and salt. And all of these nerves absorb this, and so they have their food in a way. So for me, that is the reason that the choroid plexus keeps the fluid in the ventricles clean and replenishes its oxygen and its glucose, because its job isn't in the ventricles because there's no cells to feed there. Its job is when it leaves the ventricle, it's the food for the cell, for the neural tissue as it's leaving in the form of a nerve from the spinal cord, also from the brain stem as the cranial nerves, but there's an added extra bit. Well, I'll talk about that now anyway. Here's a picture of the cranial nerves leaving. The cranial nerves, their nerve root goes through the brain stem, and the roots all nestle up against the fourth ventricle. So the root of the nerve can absorb oxygen and different nutriments to take on its first part of the journey to the face or down through the body. And these nerves go for quite a way before they go out through the dural sheath into the face. And then also from higher up in the fourth ventricle than the bottom one, there's another foramen, and cranial fluid comes out of this and then bathes that area. The cranial fluid from the fourth ventricle has oxygen, it has glucose, it has salt. So the cranial nerves that are bathing in the cranial fluid as they come out and as they travel to the face or their destination, they can receive the nutriments they need. Also the light blue area, this is called the tentorium. It's a sheet that divides the upper and lower brain. All of the cranial nerves come out in the lower brain area, it's around the brain stem. And this sheet, in a way, it stops the oxygen and things escaping away above that. All the nerves are below that, and the exits from the fourth ventricle are below that. So all of these nerves get their nutrition, and then the vent at the bottom of the fourth ventricle, this comes down and bathes all of these nerves. So for me, this is nutriments for the nerves. And also the ventricle coming right down the middle of the spine. This is the same as the cranial fluid in the body, the central nervous system, except it hasn't been used. It still has all of its nutriments, and again, the nerve roots come up around this, and it's extra nutriments as they travel through this area where they don't have their own blood supply. So they're both cranial fluid, but one keeps it, and I think the use is for the nerves. So here we have the nerves coming out. You can see the ventricle down the middle of the spine with cranial fluid with its nutriments and oxygen. You can see the body of the spine. This has a blood supply. And can also feed the nerves, the cranial fluid flowing off down through the nerve. The cranial fluid in the arachnoid space, either side, this has cranial fluid from the body of the whole central nervous system, and it also has cranial fluid from the ventricles, which has oxygen and glucose. These can absorb in through the fine membrane around and travel down and feed these nerves through this space where they don't have any blood supply. Now later on in the nerve there is the blood supply, and that is the same blood supply as in the body. It doesn't have this fine filter around it, so this is letting off all sorts of things that you don't want in the central nervous system. We'll come to that in a bit more detail in a minute. And you have to have this flow going down the nerves, because otherwise all of these ingredients coming out of the blood supply further down the nerve, they can flow up into the space, into the subarachnoid space, into the central nervous system, and the central nervous system lives in this very pristine fluid, and also different toxins and different viruses can come up. So you want all those not to be moving towards the central nervous system. You want them moving away. So you need this flow of cranial fluid moving down the nerves or down the nerve fibres and around the nerve fibres to take the movement away and down, so anything that isn't meant in the nerve, in the central nervous system, doesn't go there. Okay, so we'll come to that a bit more in a minute, how that is all coming together. So it flows through, the fluid flows down the nerve from all different directions. Some has nutriments in it, some doesn't, and it feeds these nerves until they get their blood supply. So in this next picture you can see two neurons in the central nervous system, and you can see the myelinated glial cells, and you can see that there's a space between the myelinated glial cells. There's a node, this is called a node, and it's a space between the two myelinated glial cells. Now as the nerves, as the neurons and the axons from the neurons travel down the nerves, the myelinated glial cells move together, so this node isn't there anymore, and we'll explain why in a minute. And then again in the next picture we have a nerve with three nerve fibres going down it. We have the axon, and then we have the myelinated glial cells, and notice there's no nodes there, there's no spaces between the myelinated glial cells as they travel along the axon. They push up against each other, and because the fluid surrounding these myelinated glial cells, as they get into the nerve, there's a blood supply, and that will have proteins, carbohydrates, fats, there'll be viruses, bacteria, all sorts of things, and you don't want it to get into the neural tissue. The axon and the myelinated glial cell is neural tissue. It doesn't want this heavier environment, so it joins together so that that fluid can't get to the axon. And also they come together so the fluid travelling down through the myelinated glial cells can travel directly from one to the other. So now let's look at the joint. So here we have a cross section of a myelinated glial cell, and you can see the tight connections where the different layers of it are joining to the axon, and then in the outer rim at the top, this is where it's opening, the close connections that keep it sealed. But they don't keep it sealed, that's not a good word. They only allow pure water in, 100% water in and pure water out. And you can see there's a space at the end where the axon comes out. So this is actually one that is in the central nervous system because there's a node. But in the nerves, as they travel out, there will be another one pushed up against this, so there's no space, so they're pushed up so the fluid around can't get to the axon. And what we have in the connections, in where it's travelling through the nerves, is this outer layer, top and bottom. It breaks up, or coming out of that have hundreds or thousands of fibres that join together and mesh together and make this connection. Now these fibres, for me, nobody really has been able to investigate what's happening here, so this is for me, all of this is for me, it's how I see it and how it fits in with my inner journey. For me, there's two reasons for all of these fine fibres. One is to connect it so the fluids from outside can't get to the axons. The other is it increases the outside area of the cell massively, because all of these fibres have a skin around them, and that becomes the surface area. So it increases the surface area many-fold in this joint. So what you will have is the cranial fluid flowing through the myelinated glial cell, and it will come to the end and to the junction and want to move into the next myelinated glial cell. and the pure water, the 100% water, can travel straight through these joints at the bottom and then into the next myelinated glial cell through the closed connections in that. But the salt can't, the oxygen can't, the glucose can't, the other minerals it needs can't. They have to travel into the body of the cell, these folds of the cell, then come out through the folds into the body of the next cell, and then through that and then into the fluid. And because there's such a huge area with all of these intertangled fibers from the ends of each cell, it increases the area, and the fluids can flow inside these fibers and then into the fibers of the other one, because they're hollow inside, they have fluid inside, and then into the next cell. So the water can travel down through the close connections, and the nutriments and more water, because water will move through anything, more water can travel across. So you get the water travelling through the close connections, travelling through the membranes from one cell to the other, and the cells are intelligent. The cell where it's coming to, it will accept, well, it can't not accept the water will just flow into it. It will accept oxygen, accept glucose, it will accept salt and different minerals that it needs. The carbon dioxide, which is trying to come across, doesn't want that, lets that go. That will come out through the outer surface of the myelinated glial cell into the connective tissue around, where it can be taken away by the venous blood. So here we have a cross-section again of the myelinated glial cells. Got cranial fluid in the middle. Now this one is in the central nervous system because it's cranial fluid around it. And you might ask, why would it want to make a filter in the cranial fluid? Because it's cranial fluid around it and cranial fluid within it. Well, one of the reasons that it may want to do that is that it doesn't want the carbon dioxide that will be there as well, but also maybe the cranial fluid inside the glial cell, inside the myelinated glial cell, wants slightly different things. Very likely the axon in the middle may want more salt because the electrical charge is caused by salt. So maybe it wants a higher salt solution. Maybe the solution in the myelinated glial cells is slightly different. The myelinated glial cell, the electricity moving down the axon, if it's not myelinated, it moves 50 times slower than if it's myelinated. So if you've got a myelinated cell on it, or myelinated sheath, which is lots of these cells joined together, it moves, the electricity moves 50 times faster, so it gives a quicker of whatever's happening. Now, they say that is because the myelinated glial cell insulates the axon so the electricity can travel more fast through it. I don't believe that. These are all theories. You know, the theories that have gone before theories. I've got my own theory. And my theory is the reason that it can move faster when the myelinated glial cell is there is because it can travel through the water. If it's salt water in the myelinated glial cell, this is a perfect conductor and the electricity will travel through that. So that is, and it will travel through the water and the membranes, the membranous spiral going in, this will insulate that. So I don't think so much it's an insulation for the axon. I think the insulation is for the electricity travelling through the water. If it's salt water, electricity, that's one of the quickest conductors you can get. So the reason it becomes a filter within the cranial space, I think it maybe needs a different environment. It's still 99% water, but I would guess it might need a bit more salt. And that I also think the electricity travels through the water faster than it would travel through the axon, and that's how they describe it. It jumps from node to node. It comes, it kind of stops at the node and then jumps through the next myelinated bit. So my theory is it's actually moving through the water rather than the axon, the electricity. Okay, so in the next picture we have a cross-section of a nerve. So you can see a cross-section of the myelinated glial cells, and you can see the spaces in between. Now there will be a blood supply to this without the filter that's on it in the central nervous system, and it will be the same, it's interstitial fluid and the same as is in the body. So you end up with two types of fluid flowing down through the nerve and also expanding out through the outer skin of the nerve as well. Within the myelinated glial cells, this is a sealed area, and for me these are our capillaries, and with all of these, the spiral, this layer that's wrapped around and around and around, this can wrap around over 100 times, and microscopic, you can barely see these. It's the same size as the capillaries, as the blood capillaries, which will only take one blood cell through them. So they're very fine, and it's got this thing wrapped around 100 times and a layer of fluid wrapped around 100 times, or different amounts anyway. This, and also lengthwise, because you can't draw them because you need to get them in, but they're 100 times as long as they are wide. So this makes a perfect environment for capillary action. An oak tree will take up to 250 gallons of water right up to the top leaves in a day by capillary action. And in this picture you can see the capillaries, and for me not only is there the slight pressure and the cranial fluid's got to go home somehow, it's got to get back to the heart, it's travelling down these, and it's being helped by capillary action. So again, in this photograph, in this picture, you can see the myelinated glial cells, which are neural cells. They're neural tissue, and they live in this very pristine environment of cranial fluid. So there's cranial fluid in there, 99% water. From the fluid around, it has things they need. From the blood, coming out of the blood, there's salt. They need sodium, they need salt. They need more oxygen as they travel along. They need more glucose as they travel along. So they create their own environment that's the same as the cranial one, and they absorb it from around in the same way as the ependymal cells in the central nervous system absorb the environment from the blood circulation there, because the blood circulation in the choroid plexus that is feeding the ventricles also doesn't have a filter. It's completely encapsulated by the ependymal cells, so it creates interstitial fluid, a bag of interstitial fluid, and then the ependymal cells, they take the water, they take glucose, they take oxygen and take salt, and end up with this environment: 99% water, glucose, oxygen, salt. And it is exactly the same around around the myelinated glial cells we have interstitial fluid. The myelinated glial cells will produce cranial fluid out of the interstitial fluid, and also they will maintain that. They will give the any toxins or the carbon dioxide back to the interstitial fluid around them. So, and this is the same when you look at, I'm just trying to find a picture. We have this picture of, oh, there it is. This picture of the nerve coming out. We're going to have to stop soon, I'm going to have to cut this again. And through down the nerve with the myelinated glial cells. Here's a picture of just the tissue, just the connective tissue between the myelinated glial cells, and this will take the cranial fluid that has been used, that has carbon dioxide in it, that doesn't have any oxygen left, that the salt's been used. This will travel through the connective tissue, and the oxygen, the salt, the water will be absorbed by the myelinated glial cells. So we're going to have to stop this one now, and I'll join it to the next or make a new one. Thank you.